Today we are joined by Dr. Tyler Lyson, who is the Curator of Vertebrate Paleontology at the Denver Museum of Nature and Science. Dr. Lyson has done quite a bit of work with turtles and tortoises and sort of evolutionary development of different features and anatomical aspects, as well as just the sort of phylogenetics of different testodons. And we're really excited to talk to him today. It's sort of a new aspect. We haven't really covered this area yet, so really exciting. So thanks for coming on, Dr. Lyson.
Yeah, thanks for having me. Great to be here.
So, Dr. Lyson, what got you interested in turtles and tortoises, and how did you get involved in paleontology?
Yeah, I mean, I think I've always been interested in turtles. I mean, they are God's noblest creature, after all. I think we can all agree on that. But in all
Yes.
seriousness, I grew up in southwestern North Dakota, a really small town, kind of right in the middle of nowhere, but right in the middle of dinosaur-bearing rock, sort of Earth's last dinosaur ecosystem. So my earliest childhood memories are out running around on the Badlands, looking for and finding fossils. And you find lots of dinosaurs, Triceratops and T-Rex, and lots of other things. And of course, those other things we would find were turtles, bits of turtles in the Hell Creek Formation. And I'd find, lots and lots of chunks, just pieces of turtle shell, just pieces. And as a kid, I always wanted to find a complete one, a complete shell, just because they were quite rare. So that was just kind of my earliest memories of like just wanting to find, kind of almost like the treasure hunting aspect of wanting to find a complete turtle shell, and eventually finding some of those. And then I think more of my academic interest started in college, where I went to work with Dr. Scott Gilbert, who's at Swarthmore College and who studies the evolutionary developmental origin of the turtle shell. And then finally, I went to Yale, where I ultimately decided to work on, the origins of turtles, because this was a really magnificent group of animals that's still alive today. And we didn't really have a good understanding of who they were related to, how they got their shell, when they got their shell. So there are all these really big evolutionary questions that had not yet been answered. So that's, kind of my trajectory of interest in fossil turtles.
I mean that's it's real interesting, and I think it kind of starts us off in a great place. Obviously, when people think about the turtle, the first thing that comes to mind is the shell, and as you said, you've done work with sort of the development of that feature. But when you really think about the shell, it seems sort of cumbersome, and that there's a lot of things like, why would a feature like that evolve? Why would that be the case? And I'm just sort of curious, like, what have you done in the realm of that sort of area of research, and why do you think, what's the purpose of the shell, and kind of how did it come into existence?
Yeah, I mean, it is the iconic thing about turtles, right? The turtle
Yeah.
shell. It is the iconic feature. Obviously, there's other things, the lack of teeth and sort of the anapsid skull, which I think we'll get to later. But
Right.
I mean,
Yeah.
it's the shell. And of course, today, the function is largely protection. There are other functions in terms of storage of various minerals, and which helps with estivation and being able to go underwater and without oxygen for long chunks of time. But in general, right, the function now is for function, or is for protections. Excuse me. But I mean, a lot of these complex features don't evolve for their current functional reason, right? I mean, there's many things. A classic example, of course, is feathers. Dinosaurs had feathers. Relatives of T-Rex had feathers. Today the feathers are for flight, but that is not why they evolved, because again, we have all of these other features. So it's a classic exaptation, where there's a different functional reason to begin with that then gets co-opted into its current functional purpose of protection. And so with feathers, of course, we think it's sexual selection or thermoregulation that then gets co-opted into flight. And for me, I had this idea, which is part of my PhD, that the question really is why do turtles have broadened ribs? Because ribs are so crucial for breathing. And this is why the ribs of a whale versus the ribs of a snake versus the ribs of a T-Rex versus the ribs of, pretty much any animal, they all look alike. They're long cylindrical structures because it's tied to breathing, which is crucial for living. And so turtles, on the other hand, they have broadened their ribs throughout development, and then eventually they suture those ribs together to create the immobile shell. And then, of course, there's other bones that go into making the shell. But that to me is the real crucial question, or like, why broaden your ribs to begin with? When you already, when they're tied to breathing, tied to respiration. And there are other animals that have broadened ribs, and these are animals that largely that spend a lot of time burrowing, or that use their arms for digging in these big sweeping motions, these big forearm like digging motions. And if you're a big forearm digger like that, it creates lots of torsion in your body, and it's one way to counteract those forces and to counteract that torsion is by shortening the space between your ribs, the intercostal space, and putting bone there, because it just basically stiffens everything up. So this is things like with the silky anteater and other animals like that they tend to do. So I had this idea that, well, maybe these earliest turtles were burrowers. So I had the idea, but the problem with it was testing that idea. And what we needed was some fossils that that had a good postcrania, that had good hands and arms and other elements preserved to test that idea. And then in, I think, 2014, this amazing fossil was found, this amazing fossil that I think is the earliest stem turtle. A new specimen of Uninotosaurus was found that had hands, it had the arms, and because then you could look to see, like, are there other osteological correlates for digging preserved? Not just the ribs, because we knew Uninotosaurus had these really distinctively broadened ribs. And sure enough, it had really big claws on its hands. It had a really big olecranon process on its ulna. And then we did histology of the limbs to look at the Sharpie fibers. And the Sharpie fibers are just the attachment of the muscles into the bone, and we could see that sure enough it was jam packed with Sharpie fibers, and that the cortical bone of the ulna was really, really thick. Which I always like to joke, all of these things, that the olecranon process, the Sharpie fibers, the thickened cortical bone, all indicated that Uninotosaurus was jacked, that it had really big forelimbs. And one of the ideas then would be, the idea that I had was that it was a burrowing creature using its big hands, its big forelimbs to push through the substrate to burrow. It's a thing that a lot of modern day turtles do today as well, like Gopherus. It has the same type of motion, the big forelimb strokes for digging these holes. And then the question is, like, well, why burrow? Well, if you go back to when Uninotosaurus was alive 260 million years ago, it was living in South Africa, and it was living during Pangea times when all the continents were together, and it was incredibly, incredibly arid. And so one way to deal with these really arid conditions, what a lot of animals do today, is by going underground. And by going underground, you're able to have more temperature control of your surroundings. So it's not so hot, it's not so arid. So kind of it's a big question, of like, why. But so I think the initial reason for the origin of the shell was for burrowing. That was the initial impetus that spurred the broadening of the ribs. And then with the broadening of the ribs, the next big thing is you have to have a decoupling of those ribs from breathing, because the ribs ancestrally are used in locomotion and with breathing. And so you have to decouple them. And every group of organisms, I shouldn't say that, most groups of amniotes have found ways around that. Mammals, we have the diaphragm, right? So we no longer really use our ribs as the primary force of ventilation. We still use them some, Anybody who goes out and for a long run knows that their intercostal muscles, all the hapaxial musculature, gets really, really sore and tired.
Yep.
Crocodiles have the hepatic piston. Birds have the uniflow directional breathing mechanisms. Lizards, they still are largely have that constraint, which is called a Carrier's constraint after Dave Carrier, who first proposed this idea. But so anyway, I know this is a long-winded answer, but I think the original function was for burrowing, that it led to the decoupling of the complete decoupling of the ribs from breathing, because you have to have that before you can lock your ribs up into a shell, which has only happened once, right? It's only happened with turtles. Lots of other animals have shells, but they always make a shell a different way, and that's by adding osteoderms on top of the body. So crocodiles and armadillos and placodonts and many groups of animals form shells, but their ribs are still the same boring rod-like shape because they're so functional, or tied to breathing. And it's only turtles that do it a different way. And so you have to decouple the ribs from breathing. And once you can do that, then evolution and natural selection, you can start tinkering with things. And I think, which is exactly what's happened with turtles. So, and then and so it wasn't then until later then that the shell then became its current functional role of protection.
That's really interesting. One thing that's also kind of interesting to think about, for me at least, is when I was younger and kind of first looking into this area, I thought sort of logically, if you found fossils, you'd expect to see an early stem turtle that kind of had some sort of remnants of like osteoderms or something that was kind of related to that. So, but Unitosaurus doesn't really look like a turtle. So how did you sort of narrow down, sort of hone in on the fact that that could have been a stem turtle and choose to analyze that fossil?
Yeah, I mean, I'm with you, Michael, on that one. I mean, I was a full proponent of the turtle shell forming via the acquisition of osteoderms. Like, I fully expected to find an animal that had a bunch of osteoderms, because there was this long fight between developmental biologists who were looking at the development of these turtles and seeing these ribs growing, the intramembranous bone growing out of the periosteum of the rib and the neural spines, saying like the shell forms via the broadening of the ribs and the broadening of the neural spines of the vertebrae. And then you had paleontologists finding things like pariasaurs, these early reptiles that are found in Russia and South Africa that had lots of osteoderms. And you can imagine, you just have more osteoderms, and eventually the osteoderms meld with the underlying ribs to form the shell. So these were these two competing ideas. And I was really in the osteoderm camp. Like, I fully expected we were going to find an animal that had, ribs that were with osteoderms melding together. And then that all changed in 2008 when Odontochelys semitestacea was described from the Triassic of China. And that just blew my mind. I mean, because this is this turtle that has half a shell. It has a full plastron. The full plastron is formed. It has these distinctively broadened ribs that haven't fused together yet, and no osteoderms. So it was in that moment that I became a believer that, that the developmental biologists were right all along. This 120 year fight between those two camps. In my opinion, it was that fossil brought those two camps together, the paleontologists and the developmental biologists, and showed that it was the broadening of the ribs. And so then my search image changed, because before I was always thinking like you just said, we're going to find one of these early stem turtles that had lots of osteoderms and other features of turtles. That's what we were looking for. And so Odontochelys was found. This is early on in my PhD, in an incredibly formative fossil. I didn't help describe it or anything, but so then I started to think about Odontochelys. What else looks like Odontochelys? And I remembered this image in the back of a Romer's, Alfred Romer's book on reptiles. He had an image of Unitosaurus, and Unitosaurus was thought, back in the day to be an early stem turtle, but then later paleontologists were like, yay, it has broadened ribs, but we all know that the shell forms with osteoderms. Unitosaurus lacks osteoderms, therefore it can't be an early turtle. So these two questions were like intertwined, right? They were intertwined, and one was driving the other. And so now with Odontocheles, I was like, dang, like maybe Unitosaurus is an early stem turtle. So I talked to my advisor, flew down to South Africa, and looked at all the Unitosaurus specimens. It's a common animal. There's a lot of Unitosaurus specimens. There's almost 60 specimens of Unitosaurus at different ontogenetic stages. And then we just started to find more and more features in the ribs, in the body of the animal, as well as the skull of the animal, that indicated that it very likely was one of these early stem turtles. And so at 260 million years old, it would be the oldest stem turtle. And I just love it because since 2000, we're living in the golden age for the figuring out the origin of turtles, because for 120 years, the oldest turtle was Proganachilles from the Triassic of Germany. And it already had a fully formed shell, so it didn't really say that much about the origin of the turtle shell itself. So that was like 120 years. That's all we had. And then 2008, we get Odontocheles, and then I start publishing my stuff on Notosaurus, starting in 2010 up until the recent. And then more recently, Papacheles was described at 240 million years old. And I think that, brilliantly fills the gap between Unitosaurus and Odontocheles. And then even since then, there's been another stem turtle found from China called Eorhynchocheles. So now, again, just in the last 12, 14 years, we've had all of these new fossil stem turtles from the Permian through the middle to late Triassic coming on board, coming online, and really informing the way we think about the evolution of these animals and how and when the turtle formed its shell.
Well, that was really interesting. What is like the time frame from the earliest stem turtle to when we finally get to a turtle that has a recognizable carapace with full peripheral bones and everything like that?
Yeah, that's a great question. So, I mean, we get the earliest turtle with Unitosaurus at 260. It has broadened ribs, no plastron. Then you have to go all the way up to you get to Papacheles at 240, and that has broadened ribs and broadened gastralia, which forms the plastron. But it's still not recognizable, I would say, as a turtle. And then Odontocheles has a full plastron. So that's the full recognizable feature of a turtle to form, is an Odontocheles with the plastron. And then it's not until you get to either Proganachilles or there's other turtles, a Paleocursus and Proteocursus, that are all roughly the same age at around 220 million years, that you have a fully recognizable turtle shell with a full carapace, with all the peripherals, as well as the full plastron. So there's basically 40 million years, separating those from Unitosaurus to Proganachilles and these other early Triassic turtles with a fully formed shell.
What is sort of the functional significance, do you think, of having a plastron before a carapace? Seems like it could almost support the burrowing hypothesis in a way. If you've got like a gular that's kind of fit and designed for burrowing, it could help. But I seem to recall Odontocheles was more of a marine animal, so it's
Right.
not. So what do you think that the functional significance of that is?
Yeah, so I mean, if we go back to this idea that these turtles are burrowers, and that's the initial reason for broadening of the ribs, and so they're using, and there's many ways to burrow. You can be like a dog and scratch burrow, but what the burrowing that I'm talking about is with the forelimbs, and that's these big sweeping motions with the forelimbs, right? And to dig into the ground. Now that is the same stroke that turtles and other animals use for swimming. So I think a lot of these animals, there was almost a pre-adaptation. I hate to use that word, but they were well adapted for burrowing as well as for swimming. And that's why I think we see a lot of these early turtles kind of going back and forth in that interface, between, because it probably wasn't an either or. A lot of the burrowing turtles probably were very well adept at swimming as well. And so it's the same motion. So now the plastron, your question, what about it? And so I think then with that in Odontocheles, and this was an idea that other people proposed, but that it maybe acted as a ballast. Because one thing, to help the animal go up and down in the water column. And I like that idea. I think that is a solid idea, because it still can't be for protection, right? The shell, that at that point, if you're exposing the upper part above your ribs, if an animal bites you, your belly might be protected, but your ribs and your lungs aren't. So I mean, that whole protective thing can't be the case. So I think it was these early turtles were going back and forth between the land and the water, and we see that with these different groups. And I think with Odontocheles as being one of these near shore marine animals, it may have acted as a ballast to help it move up and down in the water column.
Do you think there also could have been some camouflage aspect to it? With sea turtles, that's been something proposed as like a lighter plastron. It sort of blends them a bit better for predators that are looking from the bottom up.
Yeah, I think that's a reasonable idea as well. I mean, I think I haven't really thought about that, but I think that is reasonable as well. I just don't think, because I know it can't again be for protection at that stage. That's just one thing that I'm kind of very much against, because you're exposing your back, you're exposing your lungs, and so for the protective reason. But I like that from a camouflage. Yeah, if you're higher up and from animal lower down. I could see that as well. That could be another. And a lot of these things, it might not just be one or the other. It's usually there's kind of a number of factors going on here. So I think it's a reasonable hypothesis for sure.
Yeah, and who knows? Maybe there wasn't really a functional, like you said earlier. Some of this stuff just happens, and then it kind of fits an environment, and it works to some extent. From sort of an evolutionary development perspective, too, the turtle shell is real interesting, and I've come across some stuff that talks about sort of the interplay between the chondral and the dermal bone. But for someone that doesn't really understand kind of how that's sort of a confusing thing. If you don't understand kind of what that means in context of the ossification and so on. But sort of in what way is the shell unique from sort of an osseous perspective?
Yeah, well, it's the bones that that form it. For one, the turtle shell is formed of around 50 bones, and then it's not, for a lot of these other animals, it's all dermal bone. It's formed by osteoderms, and so osteoderms are just ossified scales. So think of a scale, and then that forms intramembranously, which basically means there's no cartilaginous stage. So it goes from tissue to bone with no cartilaginous stage, versus something where like our long bones or endochondral. It's a different type of ossification. And so the turtle shell, I think, it's super unique because if you look at the earliest beginnings of the shell, it's from an osseous perspective, it's if you look at the cross section of a rib, normal ribs are just going to be nice and round, endochondral bone, and then you get these little spindles, these little spicules of bone growing up out of the endochondral or the perichondral collar of the rib. There's intramembranous types of ossification, and that's the broadening of the ribs. You see that in the vertebrae as well. And so you have a unique blending of endochondral, which is again cartilaginous elements with the ribs and the vertebrae, as well as the unique mode of sending bone, intramembranous bone, out of that perichondral collar of the rib. And then the shell, you also incorporate lots of other elements. So you're incorporating the shoulder girdle bones. So in the plastron, you have your clavicle, as well as humans don't have an inner clavicle, but other animals do, an inner clavicle, and the clavicles go down into forming that front part of the plastron. And then there's another part, the nuchal bone, that's also formed by these shoulder girdle bones, these dermal ossifications. So again, no cartilaginous intermediates. So the shell is just, it's incredibly unique. It's 50 bones. It's formed of ribs, vertebrae, a splitting of the shoulder girdle, some of the bones going down into the plastron, some going into the carapace. And then at the very end, the very last stage that we see both ontogenetically as well as phylogenetically in the fossil record is then the appearance of all of the peripheral bones around the outside. And those are likely modified scales, modified osteoderms. So you kind of have it all. I mean, most animals again form just using osteoderms. Turtles do osteoderms plus endochondral bones plus dermal bones. So they're using kind of all aspects of the ossification scale, which was your initial question, as well as just different types of bones to form their shell.
That's it's interesting to think about the homologies of the shell and where that kind of comes from. I mean, at this point, it's bringing to mind something where I've thought about this. I spent the first semester in college reading a book about it. It was a bunch of different perspectives on the evolutionary debate, and they had sort of the creationist side of things, the intelligent design side, then sort of the more natural selection side. And it kept coming to mind that one of the flaws, or maybe not a flaw, but one of the aspects of the natural selection based mode of evolution that the intelligent design group sort of pushed back against was the fact that nowhere could anyone highlight a fact where a large jump in morphology occurred in the fossil record. And I think that that's likely not the case, but some people have proposed that turtles could be a great example of this, but it seems like maybe it was more, the change was a bit more gradual than maybe an evolutionary development perspective would have you believe. I mean, what do you think about the prospects of sort of that hopeful monsters kind of development of turtles? Is that likely, or was it more gradual?
Yeah, I mean, I think that's an interesting idea, and it's certainly one that a lot of creationists latched onto. Because again, for 100 years, the oldest fossil was Perganocheles, had a fully formed shell. So you had people saying, like, look, you have sort of these lizard-like things, and then all of a sudden you have a Perganocheles and a shell. Like, that's a huge and very fast jump. So therefore God, therefore hopeful monster. I mean, there's many jumps there because we didn't have a good fossil record of these earliest stem turtles. But man, has that changed, right? Since the discovery of Odonocheles in 2008 and the description of it in 2008. Now we just have this whole array of these more, transitional or intermediate types of morphologies. We have things that have broad ribs with a full plastron with Odonocheles. We have things with broad ribs and broad gastralia and Papacheles, and then we have things even further back. So I think that whole idea, we can kind of throw that out the window, in my opinion. We don't have to invoke the hopeful monster or any sort of like big giant step, evolutionary step to explain the evolution of the turtle shell, because we now have the fossils, and we also have kind of the understanding, or a better, I should say, we have a better understanding of the timing of a lot of this, and that it occurred, we talked earlier that it was 40 million years separating Unitosaurus from a fully formed shell. 40 million years, that's a long time, right, for natural selection to do sort of its regular things. And so we don't have to, again, invoke this idea that turtles are hopeful monsters, in my opinion. And we're only going to find more fossils, right? We're only going to find more of these intermediate fossils. It's sort of the same thing was invoked back in the day with birds and flight. How do you get to animals like that? And of course, since then we found thousands of amazing intermediate morphologies between dinosaurs, theropod dinosaurs, meat-eating dinosaurs, and birds that show, again, feathers and how they evolved from these simple structures to these, over millions of years, more complex structures. So we had similar arguments then. There's similar things have been, trying to, be invoked for turtles. And I think it's just a lack of the fossil record, which is now getting filled in. So it's really, really exciting time to be a turtle paleontologist.
That's I think that's well said. And it seems like that sort of hypothesis, obviously, it sort of seems like a way to sort of make an easy explanation of the fact that we don't have enough data at the time. Sort of just saying that this probably happened really rapidly and we can't see it, rather than it happened gradually. We just have more data to fill in. And that sort of seems like what the case is. And it's cool to see that, I mean, 20 years later, how that's actually kind of played out. So something you touched on earlier too was the breathing mechanisms and the ventilatory apparatus of turtles, and that was initially that paper I remember reading it, and just everything about that was awesome. I mean, the graphics are amazing and everything, and it sort of makes sense. It's well done. But
Thank you.
maybe you could just go over kind of how the ventilatory aspect of or apparatus of chelonians works. I mean, it's really fascinating. It'd be interesting to hear kind of how you explain that.
Yeah. So turtles, we'll start with what we know with the living turtles, and turtles have a unique ventilatory apparatus, and it's a very, very simple apparatus, which is awesome. I mean, so basically, turtles have a shell, and then they have their lungs between the shell and then all of their guts and whatnot, and then they have a muscular sling that goes from the front of the shell all the way back to the back of the shell. And when that muscle contracts, it lifts up the intestines and pushes them against the carapace, and the animal exhales. And then there's another muscle in the back of the shell, a cup-shaped muscle that's attached to the bag-like muscle. And when that muscle contracts, it flattens out, pulls the bag down, and then turtles can breathe. And so there's these two hypaxial muscles really that drive turtle respiration. And so that's been known for a really long time, that this is the way in which turtles breathe. Very simple. It's not not complex. Turtles aren't the most aerobic animals, right? They're not running around doing crazy things or flying. I mean, they're remarkable in many, many respects, but in terms of their metabolism, they're not that remarkable, I guess. And in the upper end, they're remarkable in their lower end, which we could talk about later. But so and so we know turtles don't use their ribs in lung ventilation. They're the only group of amniotes that no longer use their ribs in any aspects or any respect in lung ventilation. But because even mammals, like we still use our ribs a little bit. It's largely driven by our diaphragm. We talked about crocodiles and whatnot. So anyway, let's start. So that's what with modern day turtles. And so now, what's the ancestral condition of breathing in amniotes? And this is again something that other people have studied and that we know. But the ancestral way in which amniotes breathe are by using their ribs and then the muscles around their ribs for breathing. And so there's a dual function then for these muscles. One is for locomotion, because the ancestral animals were kind of lizard-like. Let's just think of them as lizard-like, and they're walking like this, right? And so they're going back and forth to increase their stride length. And so those muscles, those hypaxial muscles, those muscles basically your core muscles. When you're doing core, you're working your hypaxial muscles. They're involved in locomotion, and then they're also involved in breathing. And so there's a dual function. And what that means is you can only do one of those functions at a time. And so this is something called Carrier's constraint, after Dave Carrier, who proposed the idea in 1990, I believe. And so that's problematic, right? Because it's beneficial to do both, to locomote and to breathe. at the same time. And you can imagine evolutionarily, when one group figured that out, that there's an arms race to catch up, if you will. So every group except for lizards has kind of figured that out, of decoupling those locomotion from breathing. But lizards, so if we go back to lizards, they still kind of have the ancestral condition. So they are able to do some gular fluttering, and they are able to do some other things that allow them to locomote and breathe a little bit. But for the most part, most lizards can only do one or the other, run or breathe. So that's why when you're chasing a lizard, it's holding its breath because it can't, it's using all of its muscles for locomoting. And so its strategy is either to go up a tree or down a hole to escape. And then other animals have figured out ways of decoupling these things. And again, crocodiles, they have the hepatic piston, where their whole guts kind of move back and forth, being driven by this muscle that's attached to their pelvis and attached to their liver, and that's what's kind of driving the changes in pressure in their thoracic cavity. And then birds have figured out another way, and then turtles figured out an even a separate way. And it's and with them, it's using these muscles. And so I think there again, sort of the evolutionary story that I've come up with there is that turtles initially were burrowing, and with those big strokes, which we talked about earlier, and it was creating torsion. And so what was happening then, those ribs were broadening, shortening the intercostal space, the distance between the ribs, and that was creating this rigid rigid core, if you will. And then there was a dual function of the ribs and the muscles ancestrally, which we just talked about, to a division of function in turtles, where the hypaxial muscles are driven purely into a ventilatory role, and then the ribs are driven into a purely structural role. And that happens over the course of course, tens of millions of years. And then the way we tested that was by looking, for when did the lung ventilatory mechanism system in turtles appear. And the way we did that was by cutting up a lot of ribs of this animal, Unitosaurus. And because turtles have a very unique hypaxial musculature system. Again, they have the bag, but they don't have any intercostal muscles between the ribs. And so, because it's all formed by bone. And so if you look at the histology of a modern turtle, you can find Sharpie fibers here in the front, no Sharpie fibers in the middle, and then Sharpie fibers in the back. And again, Sharpie fibers are just the fibrinous insertion of muscles into bone. So it's a way for us to kind of see muscles in the skeleton of these animals. And so they have a unique histological signature, and that signature is identical to that in Unitosaurus. So we could see that Unitosaurus already at 260 million years ago seems to have had a ventilatory mechanism like that of modern day turtles, in that they lacked intercostal muscles. There were no muscles, basically in between their ribs, and only had fibrinous, a muscle in the front, a muscle in the back, which to us suggested it had a nice muscular bag. That's again one of the primary mechanisms for breathing in turtles.
That's some interesting reasoning, and it's cool that you bring all those pieces of evidence together, and it's kind of abstract, but no, abstract wouldn't be the word for it. It's sort of deductive reasoning, I guess, would be the case. It's really interesting. Yeah. And you mentioned metabolism, and that's one area where I'm pretty interested in that, and just the like you said, the low rate of turtles is incredible. I'm curious where they have a lot of adaptations physiologically to deal with the anoxia tolerance and low temperatures. Where did those adaptations come into play, and how does that interact with the evolution of the ventilatory apparatus?
Yeah, I mean, so a lot of modern animals that are living in burrows, that are living underground, they have those same adaptations for living in environments that are low in oxygen, because if you're living in a burrow and you're respirating in your burrow, I mean, it creates very low oxygen conditions. So a lot of animals that live in burrows have very, very low metabolisms. And so I think a lot of these features that sort of define modern day turtles all of those can be traced back to Unitosaurus and Pangea times at 260 million years ago. Again, where there is a big advantage for living underground. There's a lot of environmental reasons, and that's just because the environment was very, very harsh. I think it would be analogous to the middle of modern day Australia. Very, very hot and very, very arid, very difficult to survive. And then a common mechanism or common lifestyle, I guess, for a lot of animals that live in those conditions, these arid conditions, is a life underground, either underwater if there is water, but in the middle of Australia, there's not a lot of water. And so a lot of animals then go underground. And so I think that's where you can see sort of the earliest start for the crazy metabolism that we see with turtles today, where they can live, in anoxic environments for 24 hours or longer. They can go underwater, right? Modern day turtles can go underwater, under the ice, like up in Canada, when they excavate for the winter for up to 6 months. So just they can shut down their metabolism for that long. And so I think a lot of those initial features, like obviously those are highly derived things, but a lot of the beginnings of the metabolic aspects can be traced all the way back to this animal, Unitosaurus.
That's really fascinating to hear that it's from like medical perspective that I've done some looking into that, and it's just a lot of the adaptations to lowered metabolism, they sort of fit into the aging and that sort of camp and field of study. But also it seems like there's a lot of untapped potential for biomedical discoveries, and you have sort of parallel adaptations for sugar regulation, diabetes, and that sort of thing. And that's real interesting to me personally, those implications. So to know where that comes from, and you think that it happened more on land and then became more developed as radiations occurred into the water, and it probably maybe scaled back, and then it changed over time.
I think utterly fascinating. I completely agree with you. And then there's so many other aspects to that. I mean, that has allowed turtles to be so incredibly successful because of their low metabolism. They have been able to, the low metabolism, and I guess their lifestyle, either underground or underwater, have helped allow turtles survive 3 of Earth's last mass extinctions. So turtles have survived 3 of the 5 mass extinctions in Earth's history, and a lot of that we think is because of, again, their very, very slow metabolism. The fact that a lot of turtles during the end Cretaceous times or the end Permian times were burrowing. So other researchers have proposed that animals that survived the end Permian mass extinction, which is the mother of all mass extinctions, where 95% of life goes extinct, there's a higher propensity of animals that burrow to survive that mass extinction than animals that don't burrow. And so turtles likely were burrowing, and so that's probably why they survived that. And then we fast forward to Earth's last mass extinction at 66 million years ago with the extinction of the dinosaurs, and where 75% of life on Earth goes extinct. And it's for similar reasons why we think that turtles survived. They were underground, underwater, when the asteroid hit Earth, creating that thermal pulse that baked the surface of the Earth. So if you weren't underground or underwater, then you were essentially broiled. And then their slow metabolism allowed them to kind of hunker down, estivate, and weather out the nuclear winter that resulted with the blasting of parts of modern day Mexico into the atmosphere, blocking out the sun, hindering photosynthesis, knocking out the ocean systems, the plant systems. And so then turtles, turtles were the winners of all of these mass extinctions, and the commonality, and then it all comes down to their metabolism, I think, and their lifestyle of where they were living, essentially.
Yeah, and these mass extinctions themselves could have acted as sort of environmental impetus for natural selection to act on increasing metabolic capacity or lowered metabolic rate. It's interesting to think, like, if in the future there are treatments for certain illnesses developed out of parallel pathways related to that evolution of anoxia tolerance, that sort of thing, you can almost say that mass extinction is the reason that it could have played into the fact that medical treatments. It's kind of an interesting comparison.
Yeah, for sure.
Yeah. Well, I think something else that we can sort of transition into a bit is just the whole placement of turtles. Another fascinating aspect of them is the fact that it doesn't seem like we really know, paleontologists don't really know yet where they belong on the tree of reptiles and sort of abroad. So what do you think is the most likely scenario? Where do the turtles nest?
Yeah, I mean, what a question, right? I mean, that was one of the reasons why I went and studied turtles, was because it's crazy. I mean, well, one, they got this crazy morphology in the shell, and we've just talked a lot about their shell. But here we have this major group of animals alive today, and we still, with all of the technologies that we have and all the data sets that we have at our fingertips, we still don't have a good understanding of who they're related to. Who among modern animals are they related to? And so if we just look at analyses that have been published within the last 10 to 15 years, turtles have been placed on every possible part of the amniote tree of life. And that's just mind-boggling, right? They've been placed next to crocodiles, next to birds, next to the clade of crocodiles and birds. They've been placed next to the clade of crocodiles, birds, and lizards. They've been placed next to lizards. They've been placed kind of all over these things. And I've published a fair bit of conflicting data, conflicting placements on where turtles go. And it's because different data sets give you a different answer. And to me, that's really, really interesting, because there's one history of life on Earth. So some of these data sets have to be wrong. And so to me, that's a really interesting question. So I know I'm rambling a little bit here, but I just find this question so fascinating. So right now, where do I think turtles go? I think turtles are sister to archosaurs. I think turtles are sister to the group made up of crocodiles and birds. Now, none of my analyses have ever bared out that answer. Well, only one, I guess, using some of these things called microRNAs. But I think turtles are nested within diapsids. If your ancestor, or the older ideas, they thought that turtles were anapsids, nested outside of the animals with two holes in their head. But I think we've been able to show that in turtles is secondarily a derived condition, the anapsid condition, the condition where they have no holes in their heads. And so I think that they're sister to archosaurs just because there's been so much molecular data that have been published showing this result. When I first started grad school, it was like maybe one or two genes that were showing this. So we're talking some pretty big genes, but now we're talking whole genomes, not just isolated genes, but whole genomes. And we're starting to find more morphologic data that suggests that turtles are nested within diapsids, and then there are a few characters that may even put them up next to archosaurs. So I think it's still up for debate, and I think a lot more work needs to be done on this question, and it's a question that I myself, as well as colleagues at Johns Hopkins University with Dr. Gabe Bever, we're still actively trying to figure this out and try to bring the data sets together, because that's ultimately what we want. We don't want to sit here and say my data are better than your data, That's not very satisfactory. We want to kind of have the coming together of these data sets, the congruence of the data sets, like we now have with the origin of the turtle shell because of the discovery of Odontocheles. Because before it was evolution or developmental biologists arguing with paleontologists. Now because now we have the fossils and the developmental data coming together to tell the same story on the origin of the shell. And that's what we're trying to do now with the morphological data as well as the molecular data. We haven't gotten there yet, but we're working on it. So, but long answer, I think they're sister to archosaurs, and we still haven't been able to show that these combined analyses, but we're getting closer.
It's cool. It's real cool to see, kind of, as frustrating as it must be. It's sort of one of the biggest puzzles, and it's like trying to fit all the pieces together. And the genetic data, in the way I think about it, at least in my mind, is that the genetic data, if you've got a full genome, you should be covering at least contemporarily. And if you've got fossil genomes, that sort of thing, you're getting historic data, but that should cover all of the things that you would see from an evolutionary development perspective or just a paleontological perspective. It's capturing all the variation, and the similarities should pretty much fit what the other lines of evidence will fit. I mean, is that kind of the right way to think about that, or is there some nuance there that's missing?
Yeah, well, I think, generally, yeah. I mean, I think the challenge with a lot of the molecular data is that you're only sampling the tips, right? You're only sampling what's alive today because you can't get genomes from Odontocheles, unfortunately.
Right.
So you're just sampling up here, and what we're trying to figure out are these evolutionary relationships that go back 260 million years. And in some cases, we're quite rapid, right? I mean, the splitting off of turtles from other archosaurs may have been very rapid, around these mass extinction events. So figuring out origins of groups is always very difficult and always very messy. Because a lot of times these origins happen again after the mass extinction, where groups are diversifying, radiating very quickly. And so in order to capture that and see that, if you will, with molecular data is really challenging. And then with molecular dating, you're modeling it and you're trying to find the appropriate genes that are evolving at the right evolutionary rate to capture something that happened again 260 million years ago. Because there's other genes that are sort of turning over, if you will, at a much quicker rate. They're evolving much faster. Like RNA is not a great thing to be looking for, and using to be figuring out deep, old evolutionary relationships. So instead, you want to find other things that are much more slowly evolving genes, where they haven't completely erased their nucleotide sequences data, again, if you will. So it's really complicated, right? I mean, it's really complicated. And on the morphology side, we just want to find some good features that unite the different clades together. And the first step on that was figuring out what some of these earliest proto-stem turtles would have looked like. And so we've come a long way now in the last 10 or 15 years with Odontocheles and Eorhynchocheles and Papacheles and Unitosaurus. Like all of those are shedding light on what these earliest animals look like, these earliest turtles. And now we want to compare these early proto-turtles with these earliest archosaurs. And so I think we have a good understanding of what these early turtles look like right now. And we have a less good understanding of what these early archosaurs or early lepidosaurs would have looked like, because the fossil record isn't as good for either of those groups. So, so both data sets are challenged, I guess. That's the short of it.
Right, still a lot to sort of uncover there. You mentioned kind of the molecular clock analysis thing. That's something that's kind of cool to think about. But like you said, a lot of pitfalls with that. And you've done some work with like fossil calibrations, like time calibration. I'm sort of what some of the pitfalls with that are, because it seems logistically like the statistical front or the distributional frameworks you use for that. I've used like Bayesian inference for modeling ecology questions, but kind of how that fits into paleontology is sort of foreign to me. I'm curious kind of how do you do that? What's kind of the logic there?
Yeah, and I'll say that I'm not an expert in the analysis side of things. So any of the times where I've worked with figuring out divergence time estimations on modern turtles or in reptiles in general, I always work with a specialist who's very knowledgeable in those arenas in the Bayesian and beast type of analyses. And so there's a number of challenges on just the analytics. And I really am not the person that can speak to that. But then there's also the challenges on my end, on the paleo end, because you need to calibrate your molecular clocks. You need to calibrate those with fossils, to help figure out, the rate of these molecular clock, to help calibrate the molecular clocks to then figure out the divergent states. And the challenge is you want is the spottiness of the fossil record. So anytime that I've worked with different folks on this, it's figuring out like what are the best calibration points and then the ages. So you need the fossils and then you need those rocks, the fossils in which the fossils are found. You need those rocks to be well dated. So that way you can again help calibrate. And so it's finding good fossils that you can say like, yes, this is the earliest crown snapping turtle, not on the stem, but a crown. And so again, that's the challenge of finding good fossils. And so I worked with Jim Parham and Walter Joyce, who are both fossil experts. And we came up with a very robust fossil turtle calibration points, so that all molecular geneticists could use those. And our goal was to come up with the best ones that we all of us really agreed on, because there was a lot of some that were very controversial of whether this was the earliest like sea turtle or things like that. And the idea was that when we died and we're at the gates of turtle heaven, that the turtle god would be like, yes, this was you guys did us a solid on this. And so that was a sort of our internal joke. And then we teamed up with a group at the University of Bristol and in England, and there they were the real brains behind these, as well as Jim Parham, I should say, were the brains behind these, setting up these different studies and figuring out when turtles diverge. And so one of the big questions is like, when did modern day turtles diverge? We've been talking about stem turtles or proto turtles, but when was the modern radiation of living turtles? When did that occur? And sort of there's these different hypotheses of it was in the Jurassic or it was all the way further back in the Triassic. So separated by some 30 million years or so. And our analyses and I think they're pretty well accepted because we published these, and I think in 2011, 2013 have all been pretty well substantiated and showing that the major radiation of turtles seems to have occurred in the earliest Jurassic. So
Yeah. It's
an exciting time because to be for paleontologists, because a lot of our data now are being used by molecular biologists in figuring out divergence point estimations, developmental biologists who are tinkering with genes and tinkering with the timing and placement of transcription factors or genes or this or that to try to figure out how the turtle formed its shell. But with that, you can it's almost like Frankenstein type science. And I say that in the nicest way possible, but we need the fossil record to then go back to say like, well, we can change, we can create this morphology using, by changing these sequences or changing the timing of these, or the placement of these genes or transcription factors but did that morphology actually ever occur, right? Or is this really just Frankenstein type science? And the way, we can look at that is by looking at the fossil record and saying like, oh yes, we do have an animal that has broad ribs but doesn't have a plastron, or we do have an animal that has broad ribs and a broad plastron. So, I think again, it's an exciting time because our data of the fossil record data is becoming very, very useful now for molecular as well as developmental biologists. We're all working together now to help answer these really big questions.
Jack, are you saying something? Oh, it's
So I'm trying to avoid how to phrase this, but I was where do the like myeliniforms like fit into all of this, like in like comparison to modern turtles? Like that's been kind of unclear to me for a while, and I've wanted to know more about that.
Yeah, these myeliniforms are these awesome turtles that lived in Gondwana. So they lived in the southern hemisphere. Found some amazing fossils in Australia and Lord Howe Island. Juliana Sterley's been finding a lot of amazing these fossils down in Argentina. And these are these crazy, horned turtles, right? I mean, really, really cool animals. They have tail clubs, big bison-like horns, right? I mean, they're not bison horns, but I mean, they have these big projections. Anyway, some of the coolest turtles that ever lived, and the group, went extinct not all that long ago in the Pleistocene. And the thought with these is that they're all stem turtles. So there is some debate. Like Gene Gaffney, who's one of the big main researchers of fossil turtles, he put them in one part of the tree, higher up in the crown. But he was also kind of on the fence if you really chatted with him. But all of the recent analyses place the myelinids on the stem, kind of this evolutionary lead up to the modern crown. So they're stem turtles. They're stem turtles, not as basal as like Perganachilles or Paleocursus or Proteocursus, higher up than that, but still really, really basal. That almost made it to today, right? I mean, that's what a shame because they're just some of the coolest turtles that ever lived. They would have been like the monotremes of turtles, right? Because the platypus, right? That's a group that's almost went extinct. There's only three, right? Three monotremes, but they say so much about the evolution of mammals, and they're so important. And I think had we had the myelinids, alive with us today, I mean, they would tell us we would. Maybe we wouldn't be here trying to figure out the origin of turtles, for example, of where turtles go, because I think they could tell us a lot. So anyway, cool group of turtles. Wish they were with us.
That question pretty well.
Almost.
Definitely keeps me up at night knowing we were so close to just being able to see them out in the field.
Yep. I know. I always wonder, like with, can we pull genetic data from them, and with the CRISPR technology?
Right.
This is not my area. I'm a paleontologist, right? But is there any chance? I'm sure at some point we're going to bring back the woolly mammoth.
If we could bring back the woolly mammoth, it seems like it'd be feasible to bring back some myelinids.
It does, right? I mean, again,
put them in
I'm not the
Pleistocene
expert
Park.
at all. But,
it's crazy because humans and myelinids did coexist at some point in history. It just blows my mind. Like in the the grand scheme of things, it's like super duper close. We missed them by a hair.
Exactly. Yep. Likely eaten to extinction. Right.
Yeah. Just like the Hesperotestudos.
Yep. Yeah, I mean, it's wild. I mean, I know we're wandering here, but turtles have made it through these the last three mass extinctions largely unscathed. This is always an important point I talk about when I do my tours at the museum or when I do some of my turtle talks, is that turtles do really well. We don't really know how well they do across the Permian Triassic because there's not a good fossil record, but they are the winners at the Cretaceous Paleocene mass extinction. They do the best out of any of the amnio groups. If you only looked at turtles, you wouldn't know there was a mass extinction then, when this giant asteroid strikes Earth. But now turtles are one of the hardest hit groups, which is why we need more people like the three of you guys and anybody who's listening here, people interested in the conservation of turtles, because turtles are one of the hardest hit groups now among amniotes. Because they are so well adapted for dealing with these natural disasters in terms of asteroids and climate change and this and that, but they're not that well adapted for humans, essentially, and the overpopulation of the world by humans. And so, right, I mean, biolanids likely went extinct because humans were eating them, and that's just really sad. And there's a lot of other examples, Wyatt, that you had just mentioned. There's a lot of examples of that. It's just a shame. Very sad.
Our tortoise diversity got hit so hard, and it is just amazing to think of it. That I was going to say something about it earlier when how robust turtles are in general at getting through all of these natural mass extinctions and just trucking them off while the rest of the animals are getting hit much harder. But then we're like the biggest challenge. We could potentially be the biggest challenge they've ever faced, depending on how the next few centuries go.
It speaks to how unnatural this sixth mass extinction that we're very likely entering into is, right? I mean, I think that really just underscores it. It's just not like any of the other mass extinctions. And I mean, to be fair, each of the last mass extinctions have all been different. This is just the next iteration. I am optimistic because I know, I mean, like you said, the tortoises are getting hammered, and there's other groups of turtles that are really, really getting hammered. And there's only 356 species to begin with. So when you cut it, you take out a few, you're really cutting down percentage-wise on the number of turtles. But I am optimistic that turtles will survive this, as the group as a whole. It's just that the diversity is really going to suffer. But there are some turtles that just do really well, that are able to habitate with humans, quite well. But still, need to do more to save these magnificent animals. That's for sure.
Yeah, I think that's a good point. It's that turtles. I don't think we have to worry about turtles as a whole disappearing. But it's how much diversity are we going to lose? Is really like.
I agree.
I think the rated slider's going anywhere, but there are dozens and dozens of other species that are like right on the cusp of like where we're going to lose those lineages most likely, and some of them we've already lost. So
some of the giant soft shell turtles, right?
I have to go soon to go to work, but I just wanted to squeeze in one last question from me right before I leave. Jack and I have, since we were speaking about Hesperotestudo and burrowing earlier, Jack and I both have this little hypothesis that maybe Hesperotestudo could have been a burrower, much like Gophorus is now. What are your thoughts on that? Do you think that could have been a possibility?
Yeah, I mean, I think it has. I think a lot of the modern day tortoises, they're burrowing animals, and obviously it's very big, but I mean, it had big giant claws. I mean, I think it was probably. I think that's a solid hypothesis.
Because I mean,
well adapted for burrowing.
Yeah, because I mean, they lived in a similar with similar megafaunal mammals, so like mastodons that the sulcata tortoises in Africa deal with today. So I feel like they could have filled a very similar niche.
Yeah, absolutely. And there's always a spectrum from barely being able to dig to being a very well adept burrower like Gophorus, right? I mean, being one of these keystone species, and so maybe Hesperotestudo was somewhere there, in the middle. So, and figuring that out, like even with the Cubotosaurus, this is something I've thought a lot about, is trying to figure out like where on the spectrum, but it's really challenging to figure out where on the spectrum. You can just simply sort of list all of the different osteological correlates that these animals have, either for digging or for counteracting the force and the tension created by digging. And so And I haven't done that. I've done that for like Gopherus and Unitosaurus, and made the comparison that those two were very, very similar. But that's something you might be able to do to test that idea. Just to see like how many correlates are there, and how do they match up to something that's really that everybody knows is a really adept burrower.
All right, cool.
Yeah, I hadn't particularly considered. Just looking at the bones I've collected is kind of how I was thinking of that, but I'm pretty sure that conclusion's been thrown around and talked about for a long time with that genus. But just seeing it for myself really was like kind of made me pick the side of like, yeah, these had to have been boars. I mean, the forelimbs, the shape of the shell, the massive like osteoderms, and it's such a similar build to a sulcata, like you were saying. I mean, it wouldn't make total sense if they were burrowers, but I think on the topic of fossils, what was one of your more interesting fossil discoveries? And where have you found fossil turtles that you've least expected?
Yeah, man. So I've mostly, most of my field work is in around the Cretaceous Paleogene boundary. So I do a lot of work and found and named lots of new species of turtles from just before the extinction and just after the mass extinction. And some of my favorite, I think my favorite locality is a locality that I named the Turtle Graveyard. And it's just this super rich locality from the Hell Creek Formation where we found, I think, over 60 specimens. And turtles in the Hell Creek are quite rare. So the Hell Creek Formation is the rock unit that's famous because it's Earth's last dinosaur ecosystem. And you have T-Rex and Triceratops, and then a whole diversity of other animals, including turtles. And turtles are not nice turtles, and skulls are just not that common. And I found one locality that's just produced a remarkable assemblage of amazing turtles, and several of which are new to science that I've named over the years. One I named a Gamera Bena. So Gamera is the fire-breathing turtle in Godzilla. So I thought that was a solid name. And that's been one of my favorite localities, I think, just because we've been excavating it for years and years, and we're finding articulated hands and feet. And then I have to mention one other one and this isn't one that I found, but it's one that I worked on. And this is one from rocks that are 76 million years old. It's from the Kaparowitz Formation. Yeah. And here we found the Basilemys turtle. And so Basilemys is a giant, big, about a meter carapace. And it's a tortoise-like turtle. It's not related to tortoises, but it's a turtle that belongs to the Nanzun Chelid clade, and it goes extinct at the Cretaceous Paleogene boundary. But it's one of the cooler turtles because it's got a big shell. It's tortoise-like in that it was eating plants. It was living on the land. It had giant osteoderms all over its body. And we found one locality in the Grand Staircase Escalante National Monument in Utah that had 6 individuals, and 2 of the individuals had eggs in their bellies. So it's something that I'm currently working on publishing, but it's just such a remarkable locality because we have these amazingly well preserved fossils, articulated hands and feet, and skulls and neck. But then, the real cherry on top is finding 2 of them that had eggs that had not yet been laid. So they had started to ossify, obviously, but they were both two gravid females in this deposit, which is really cool.
That's fascinating. That's not common. You said this was just a terrestrial deposit, right? So that just to find that, that's really interesting.
Yeah, and to capture that moment from when the eggs have started to ossify to when she would have laid the eggs, that's only about a 1 to couple week window where you would have, where you could capture that, where the eggs were ossified enough and where they hadn't been laid. So obviously very sad, but we're going to be able to say a lot about seasonality, as well as the fact that we have 6 of these turtles all together. We might see something about sociality of this group of turtle as a whole. And the group's extinct, so it's pretty cool that we'll be able to provide some key insights into environment as well as maybe the behavior of this group of turtle, which I think is pretty cool.
That's real interesting. Yeah, well, we can start to sort of wrap things up, but I've got one last sort of quick question. Maybe not a quick question, but one last kind of big question. In terms of, so you mentioned earlier working on sort of the split of turtles and the deep time stem lineage split. But in terms of more contemporary or just along the, I guess, crown tested on phylogeny, where are the biggest problems that you think that need to be resolved?
I think the biggest is soft shell turtles. Soft shell turtles, the aliens of turtles. You know, where do they fit in? I mean, obviously they're cryptodires, but even some molecular data places them sort of a trichotomy with cryptodires and pleurodires. And we all agree that they're cryptodires, but I mean, when and where did they diverge? And what are some of these early transitional fossils? What do those look like? And we'll likely find those in the early Cretaceous, maybe late Jurassic of probably Asia, because the group likely originated, you know, it seems like it undoubtedly originated in the Northern Hemisphere and probably maybe in Asia. So I think that's a really big question, just the evolution of soft shell turtles in general. I think that's a big one. So it would be awesome to find some fossils from early Cretaceous. You know, we're getting some from China. It would be nice to start finding more, you know, older and that have more of these, you know, again, these transitional morphologies. So we can talk about sort of how and when that body plan, which is really unique, even within a unique body plan, right? Already within turtles. But soft shell turtles I think, are very, very fascinating, and a lot more work needs to be done on the origin of that group. That's where, I mean, for me, that's the biggest. That's, I think, one of the bigger questions.
It's curious to think about what kind of environmental factors led to that. It's such a reduction of safety for speed. It's like takes it way to the extreme. So yeah, to understand why that is.
Yeah, so it'd be interesting to see how that occurred. Was it whole hog where they started to lose all of their peripherals, or was there a certain pattern to that? You know, this reduction of their shell, and then when that occurred, and then the environment in which they're found, so we can start to piece together that story, because I do think that is that's one of these untold stories. So, be a great project for somebody.
When's the oldest? How old is the oldest soft shell fossil that we have?
I think it goes back into the early Cretaceous.
Okay.
Yeah, like the Johol. I think there might be there's fragments that are even older than that. So I know my colleague Walter Joyce. He currently has a grant, and he's working on aspects of this. And so I know it's something that he and I talk a lot about. But we won't be able to make some serious inroads until we start to find more fossils. So that's going to be one of the key challenges. So they're out there. They got to be out there, right? I mean, it's just like, but I mean, we might have to wait many more years. Again, it's analogous to the oldest turtle, which I keep on going back to, right? I mean, 120 years, Perganocheles is all we had. And then, man, now it's just been a bonanza of new, amazing transitional stem turtle morphologies just in the last 10 to 15 years. So we're sort of waiting for that to happen with soft shell turtles.
I seem to recall too molecular phylogenies put the split when they were just considered to be a group that was separate from the cryptodires and pleurodires, like you said, the trichotomy almost.
The trichotomy.
It was at like 160 million years, and I think the oldest fossil was like 110. So that seems like there's so much more time that needs to get filled in. I mean,
yeah, and I would say that split is way too old. I mean, that's some of these challenges with some of the molecular divergence time estimations, is that they oftentimes put the estimates way too old. I mean, we understand as paleontologists that our record is imperfect, that we're not always going to find the oldest of anything. It's just the nature of our data. But our data aren't that bad where we're going to have, 100 million year gap in a lot of our, there might be occasional gaps like that for various reasons, environmental reasons, but for turtles we wouldn't expect that, just because turtles are living in environments where deposition is occurring, and so therefore we should be finding some of these fossils. The challenge with earliest turtles is that a lot of them were terrestrial, not living in water or living in other environments. But soft shell turtles, we fully expect were aquatic at least, so we should find them. So I know it's a challenge. I mean, the molecular data places them so far back. I don't think no paleontologist thinks they go that far back, and I think we all think they're well within cryptodire turtles. But I think that's interesting that the molecular data is even showing that. So there's something interesting going on with the molecular data there. That somebody that's more savvy with molecular data than I am should and could address.
And the confidence interval may just overlap. Like it's got to be something ridiculous. So just a lot of.
Yeah, exactly.
It's like a best guess, but really rough estimate.
Yeah.
Well, that's a definitely interesting turtles and a cool take on it. But I guess for a last sort of to wrap things up here, just if you had one piece or multiple pieces of advice for someone looking to make turtle and tortoise work part of a career or a career, what would that be?
Well, I think I would say that I mean it's there's a lot of work to be done. I think we need a lot more. I would just encourage anybody to go on and make a career of it. And there's a lot of work to be done, and there are a lot of amazing fossils. So just speaking from my field, and not all, extant turtles and whatnot, but just being a paleoturtleologist, if you will, we need a lot more people in the field pushing our field forward. And I think it's a good opportunity. Unlike some other areas, like a lot of paleontologists, they want to go study dinosaurs, they want to go study T-Rex, they want to go study and there's just not that many fossils compared to turtles. We have an amazing fossil record, and a lot of it is hugely understudied. And there's a lot of big evolutionary questions that could be addressed with fossil turtles because we have such a great fossil record. And it's not a oversaturated field, unlike that in dinosaur land or in anthropology, people studying hominid fossils. So it would encourage more people to consider fossil turtles because great data set, big evolutionary questions, and then I think you could go and use that to really catapult your career. And it's more relevant, I think, than studying a group of organisms that is extinct. It's one of the reasons why I studied fossil turtles versus dinosaurs, because I've also collected lots of dinosaurs. And so during my PhD, it was like dinosaurs or turtles. And I ultimately chose turtles over dinosaurs because of the better fossil record, and then they're more relevant to today because they're still alive. And I think they're just a great model organism to study. And so I would just encourage more budding herpetologists and paleoherpetologists to consider studying fossil turtles.
Sweet. That's good words. We'll have to, Jason, one of the guys that couldn't make it today, is an anthropology major, so we'll have to that's some good advice for him specifically. We'll let him know. Well, just to finish things off, we like to do a little, I think I mentioned this, just a little quick trivia volley, turtle trivia. And again, I guess the most obscure turtle facts just a way to bring in. One of our previous guests said that the knowledge that can't be used anywhere else, that we pride ourselves on knowing. So I don't know if you've got a few turtle trivia questions for us, or we can throw a few your way, or whatever you want to do. I mean,
well, let's hear some of yours. I want to see, and I doubt I'll have the answers to them, but I want to add to my knowledge. I have a few, but I'm sure this audience already knows my turtle trivia. I'm used to giving turtle trivia to the broader public, which knows nothing about turtles. I feel like this audience is pretty knowledgeable on their fossil turtles, given the name of the podcast.
yeah, I don't know. I mean, there's some niche things that probably you could pull out, but yeah, we can throw a few your way. Jack, I don't know if you've got something. I've got something I'm thinking of. I'll toss one to you. I've got one that just kind of popped into my head. It's not super exciting, but we'll see. So the map turtles and sawbacks, we were talking about those earlier, but the extant species, there's something interesting about the females versus the males. They get much larger and bigger heads. But the question I've got is, approximately, this isn't like there's not really an exact answer for this, but approximately what is the difference between the age at reproductive maturity for a female and a male? So what's the difference between the females mature later on? I'll give you that hint, but what's approximately the difference in age?
5 years.
I mean, it's close. I would say more like, based on current estimates, probably 8 to 10. But 5's close enough. And
that was a wild guess on my part. So
yeah, I mean, it's not bad for kind of a niche question, but.
So yeah, so how many turtles go extinct at the Cretaceous Paleogene boundary that we have good data for?
Yeah, see that.
So to put that in context, 90% of mammals go extinct, 75% of life goes extinct, 60% of angiosperms go extinct. So in North America, we'll just start with that. How many lineages of turtles goes extinct?
I'm thinking about this. There's a number that's popping into my head, but I don't know why. I think it's a little too high. I can throw it out there. We'll see. Maybe a higher. I'd say like 25 to 30.
So in North America, so it's one of the most diverse ecosystems for fossil turtle ecosystems. It's comparable to that found down in Louisiana, or down in Louisiana or Alabama. So there's 35 or so species of turtles in North America, in this one drainage system, in the Hell Creek drainage system, if you will. And we only have evidence for 2 species going extinct across the Cretaceous Paleogene boundary, which just underscores how well they did, right? Dinosaurs wiped out. Non-avian dinosaurs, 90%. Lepidosaurs, lizards, 90%. And turtles, it's like two lineage, or 2 species, Helipanoplia and the Basilemes, this big land tortoise-like animal. So
like 6 or 7%, something around there.
Right. Exactly.
Wow.
Yeah. It's basically background extinction. So you wouldn't even know there was a mass extinction, which is wild. You wouldn't know a giant space rock hit Earth, right? If you only looked at turtles. Just speaks to how resilient that group is.
That's impressive.
Jack, I don't know. You want to throw one more, and then Dr. Lyson maybe throw one more at us, and we can wrap stuff up? It's always tough getting these on the spot. I'm trying to. I had one.
So I always have one, and it's not my work, but I mean, I just love that the turtles are able to slow down their metabolism to a point where they only have 1 to 2 heartbeats per minute. This audience probably knows that, right? They're probably familiar with some of those physiological studies that have been done. But I know when give that to the broader audience. Like that's something that folks find pretty, and that's not that obscure, but it's amazing.
Yeah, that is one. I was digging through some stuff recently too, and like they compare the top end of a cheetah versus an anoxic turtle, and it was like
Yep.
160,000 times difference. So like
Yeah.
that statistic, that's just crazy too.
Yeah. Yeah, the physiology. I mean, I think that's where you get the most amazing facts about turtles. It's just their physiology, their ability to estivate for up to 6 months. I mean, yeah, and that's all the work of what Jackson, I'm drawing a blank on his first name, wrote the book Life in the Half Shell. Amazing book. If the folks haven't read that, that's an incredible book.
Yeah, that's a good one for sure. That's all right, cool. Jack's drawing a blank, I think.
Nothing. I'm trying to think of something
You
that's
got
like
something.
really.
You got something. It's something about I don't know, terrapins or something.
It's got to be like relevant. I'm trying to. Give me a
Mine
second.
wasn't relevant at all. All right, well we can start to wrap things up. If Jack finds one in the next like 30 seconds, he can ask it, but I think he's for there he goes.
Oh,
He's
all right,
thinking.
never mind. We'll just. I'll have to. I'll give up on that one.
All right, well that's I think that's a good place to end it. Dr. Lyson, thanks so much for coming on. It's been a real pleasure, and I've certainly learned a lot. It's real cool to, yeah.
Yeah,
Go ahead.
pleasure being here. I really appreciate being on and being able to chat with fellow turtle lovers. I mean, these really are just magnificent animals, and we need more people studying basically all aspects of their biology and paleontology, ultimately to help, I think, conserve, and to save the biodiversity, their biodiversity, because they're definitely hurting. So we need more good turtle workers and turtle lovers out there.
I'm back. My manager texted me while I was on my way in. We're overstaffed, so let me off for the day.
Perfect timing.
Yeah. We're closing out now, but yeah, we just kind of did the trivia, so you didn't miss too much. But yeah, But all right, cool. Well, it's been a pleasure, and thanks for listening, everyone. And we're gonna sign off.